Welcome everybody to the W27 Metabolomics workshop at UCLA QC Collaboratory. This is an introduction, a brief overview on the topics that we will talk about today, particularly as I've seen from the roster that most of you are either approaching metabolism studies or new to this type of studies. Today, as a first day, we will go through the foundations of metabolic studies, like really to understand what are what is the context, both in the technology, but also in the science and application, and what type of questions we can answer using this really an entry-level understanding of how to analyze the data and interpret them from a really NOIAB standpoint. In the next 2 days, we will go more in detail using our studio and more complex workflows. So for those that want to get deeper into this topic, there is going to be plenty of I would say a bit more less entry-level examples, but really today is these 3 hours are for you to feel comfortable in understanding the definitions,
You're muted.
Okay. Sophia and Anadita, can you hear me?
Yes, I can hear you.
Okay. We had a technical issue with Zoom.
But I can't see your screen.
Yes, okay. Okay, we will restart it because we need to record it. Okay, good morning to everybody. Welcome to the W27 Metabolomics workshop. We will have 3 sections in today's workshop, 45 minutes on introductions on systems biology of metabolism and techniques, break, then we will go deeper into workflows, and finally we will go through 2 examples. Today is meant to give you foundation and understanding of the terminology, the approaches, and giving you an entry-level understanding of how to analyze metabolomics data sets using simple graphical interfaces. Tomorrow and the day after, we will go more in depth into more complex workflows. Okay, while I go through the workflow, if you're welcome to connect to this Dropbox folder, where you can find and download the examples that we will use later today. Okay, so let's get started. Why metabolism? Why are we interested in our projects to look into metabolism, particularly from a systems level perspective? I like to start my talks showing this image. This is a stylization of the Vitruvian Man by Leonardo da Vinci. And the reason why I show it is because this was one of the first attempts in history of us looking at the human body in a very holistic way as a system, trying to find the geometric proportions, but also in the work of Leonardo, better understanding physiology and really laying the foundations of a more scientific method. So today, with systems biology of metabolism, we want to understand how this system works, and particularly what is the role of metabolism in health and disease. So do metabolites have activity, and can we use metabolic vulnerabilities to treat disease? And we are fortunate enough to be a scientist and researcher in a very data-rich and technically wealthy area for science, where we can use systems biology, so the tools of omics and bioinformatics, and particularly metabolomics. But why are we so interested in metabolism? So metabolites lie at the interface of our system with the environment. We not only metabolize these compounds to generate energy, we use them as building blocks, but in the context of our system, they are actively modulating the functions of proteins, of RNA and of DNA transcription. In addition to this, when our system is interfacing with the environment, for instance, through the diet or through our microbiota, our microbiome, this communication, this two-way communication is performed through metabolites as well. So they are really key in understanding how the system works and in dictating the phenotype. And when we talk of phenotype, we can think of the whole system, but as we gradually dissect and go down to the cellular and molecular level, we can see that it is not only the activity of the metabolites of the single molecule that acts at the single cell level that is important, but what really enables the function is the heterogeneity and strategic expression of the metabolic programs over space as to compose organs and as to be incorporated in a system that enables function. So really, there are at least 2 dimensions that are interesting to us. One is what is the activity, the function of these metabolites in our system, but the other one is also how these metabolic programs are heterogeneously expressed over space in tissues, organs, within the cell as well, like in subcellular distributions, and how does that enable, affect, and dictate function. So in order to answer this type of questions, we have powerful tools that fall under the umbrella of metabolomics. So, and this is, so the entry-level question is, how do we chart metabolism? Like, what is metabolism? What is the dimension of this space when we talk of metabolism? And traditionally and a bit historically from our classic biochemical classes and lessons, we are used to think of metabolism as a two-dimensional problem. So we have pathways, this are distributed two-dimensionally. We are to some extent try to chart what is known of metabolism in the sense of central carbon metabolism. So what are the core functionalities of metabolism in the cell that enable not only the cell to produce energy and use that energy, but also generating the building blocks for the cell per se. But as we see it as a classical two-dimensional problem with a map and a defined space, the reality is very different. The reality is that metabolism is far, and this map is very far from being completely understood and even just dissected in terms of biotransformations, connections between different pathways and regulations thereof, but also we do not know completely the structures and the components of this map. So there is, as I'm trying to depict in this slide with a hand that is still drawing structures, there is yet what in technical terms they call black matter of metabolism. So metabolites that are present in our system in nature as well, that we don't know their chemical structure, we don't know their activity, and we don't know how they interact with us also in the context of disease. So that's way far from being a defined space, and it's definitely not two-dimensional. So this is mostly more a multidimensional problem. Therefore, we need multidimensional techniques and approaches to model the data and understand the regulation. And in addition to this is all framed in the context of the proteomics regulatory layer, transcriptomics layer. And fortunately enough, we have powerful tools as mass spectrometry that enable us to shed light on metabolism and collect data at high throughput and then model this data. So how many unknowns and metabolic connections? This is really, I would say, one of the fundamental questions that is still open in this field. But also attached to that, what is their biological function? We know they are building blocks, they provide energy. We know some of their activities. Some of these activities have been extensively leveraged in pharmacology for developing drugs or also serving when administered exogenously per se as very effective drugs. However, we are far from having a full understanding of this space. So we do have an analytical toolbox. This is mostly based on two techniques, NMR, magnetic resonance approaches, and mass spectrometry. The one that is mostly used in laboratory is mass spectrometry, and the reason is twofold. So mass spectrometry is more of a versatile approach. It's easier to acquire it for laboratories. So really in terms of acquisition of resources is easier to acquire it for a laboratory, but also has way higher sensitivity as opposed to NMR approaches. Now, the advantage of using NMR is in that it provides identification of the metabolites that are measured while measuring. However, the sensitivity is really far from what we can achieve from mass spectrometry. That, on the other hand, the caveat is in that the identification of metabolites or of their structure, it goes through more steps. And as we will see in the next slides, is more of a especially for those metabolites and structures that are not known, requires some hypothesis and testing, and so it's more of a dynamic process, and it's time consuming as well. So we can use mass spectrometry in two different ways. The First one, and this the classic one, the targeted approach, targeted metabolomics, is when we already know in this chart, in this two-dimensional chart, which pathways we hypothesize and infer to be involved in the project or research that we are performing. And because of that, we are able to select the pathways, select the metabolites that we want to measure, and then develop on the mass spectrometer a method that specifically targets those metabolites, quantifies them, and provides us a report of abundances and identities. Now, the positive in that is that we obtain direct identification and quantitative measurements. The negative in that is that we only see what we want to see. We only see what we expect to see, and we expect to change. But as we said, this is a multidimensional problem. Many of these molecules and these biochemical connections are not known. So there is much more to understand from an unbiased approach in this field. And this is what is allowed through global untargeted metabolomics. So these are two terms that are used interchangeably, global or untargeted metabolomics. They mean the same, and they refer to the usage of the mass spectrometer in what is called full scan acquisition methods that allow to measure as many signals for molecules as possible within a defined detection window and with a specific setup, a chromatographic setup, and the experimental conditions that we have in our analysis, meaning that we will not only see metabolites and signals for metabolites that we know, but we will also collect those signals for metabolites that we don't know. We don't know structurally, we don't know biochemically, we don't know their effect in the context of the pharmacology of our system. So we will detect those molecules, and we will be able to measure their abundances, and through big data, get a report that enables us to say these molecules, together with the ones that I know, are changing. And that will allow us, on one hand, to look into these regulations of known pathways that we did not infer being changing in our project. But on the other hand, it also allows us to see patterns of metabolites that are unknown, that are yet small molecules, that we can see that the signal is consistent with metabolites that are changing in our system. And for those that have the time and resources to follow up on identification, looking into identifying their structure and better characterizing those pathways. So this is really more of an exploratory approach, both in terms of generating hypotheses, so the hypothesis generation, but also the pure discovery in terms of novel metabolic entities. So how does a global, let's focus on the global metabolomics workflow. And the reason for that is because the targeted the main differences are really in targeting specific metabolites and quantifying them. So most of the workflow beyond that is very similar to the global one. So it's incorporated as part of the global one. So how does practically global metabolomics workflow look like in the lab? We usually have our conditions of interest, right? Our A/B control treatment conditions with appropriate replicates. We use the mass spectrometer, generally in a configuration. The most common is the liquid chromatography mass spectrometry configuration. There is also the gas spectrometry, chromatography mass spectrometry configuration that is used in specific applications. However, the LC is way more versatile. So this is usually the one that is present in research laboratories. And these platforms allow to collect what are called high-resolution mass spectra that you can see in the center of this slide. And what a mass spectra is a collection of the signal that is recorded from summing up all the abundances of the metabolites that we can measure in our system at a certain chromatographic time. We will look better into this concept later in the slides as to simplify that. So, we can do this, we can acquire this information in an approach that is called the multimodal metabolomics. And this is because different types of chromatographies, different types of LC chromatographies have bias towards different subsections of metabolism. And this is because of the chemical-physical nature of the separation principle that is involved in liquid chromatography. So for this reason, there are available different columns, chromatographic columns. These all come with different stationary phases that are responsible for the separation and retention of the metabolites that we measure. We from the literature, but also from the experience in the laboratory, we do know that some allow to cover more on polar metabolites, so the polar fraction of the metabolome. Some others allow to measure more the lipid or mildly lipidic fraction of the metabolome. And what we do when we want to have a complete coverage, we measure the same samples using different setups. And that allows us, by combining the data, to have a more complete, more comprehensive picture of the map that I showed you before. Then we have, of course, data analysis and interpretation. And finally, so this is the step that is different when we talk about targeted and untargeted approaches. So the targeted approaches will perform the identification of the metabolites earlier, at an earlier stage, so right after the LCMS profiling acquisition. While the untargeted, the global approaches, the identification, the final identification of the metabolites is left at the end, after data analysis, meaning that once that what is called the metabolic features are detected, and with metabolic features, we refer to those signals from the mass spectra that are consistent with the signal of metabolite. However, at that stage, we can only say this is consistent with a metabolite, but we cannot say which metabolite it is and what is their structure, right? So the last step in the global metabolomics workflow is to get those metabolic features that show significance in changes across our two conditions and really defining what is their identity. And this is done by going back to the mass spectrometer, selecting, so focusing on those metabolic features of interest, fragmenting them by exposing them to high energy in the collision chamber of the mass spec, and collecting what is called the fragmentation spectra, or also MS2 or MSMS. And the reason why we do that is because it has been shown from previous studies that a specific molecule under the fragmentation under like exposure to energy that fragments it will break into fragments that are very characteristic of the original molecule. And by interpreting those fragments and by measuring the overall mass of that molecule, we can infer structure and we can get back to the identity of the metabolite. Any questions so far? Okay. Okay, so let's look closer into the workflow. So we generally start with a sample prep protocol, and the sample prep protocol, which really is at the level 0 of what depicted in this slide. The sample prep protocol can be tuned in different ways, and it really depends on what is the aim of our analysis. So when we aim to have a very broad coverage, then of course we will tune that sample prep, and there are many different approaches to do that. So the literature is very rich, is very wealthy in that sense. When we will want to have a broad coverage, then we will go for extractions that are less selective. But when we want to have more selectivity and sensitivity for specific subsections of metabolism, then we can tune that as well. And so the caveat here is that the more coverage you have, the more molecules you have on injecting your mass spec. And particularly, the more molecules you inject also at the same time in the mass spec, even if you have chromatographic separation that helps you separating and injecting analytes at different times in the mass spec, the less specific your sample prep is, the more molecules you will have injected in the mass spec at the same time. And that is relevant because that is causing a phenomenon which is called ion suppression. So meaning that the more you inject in the ionization chamber, in the ionization source of the mass spec at the same time, the less molecules will be ionized per type of molecule. And the reason is because you have the same amount of ionization potential for more mass, right? But it's also, there is another aspect to that, is that species that are at very high abundance tend to suppress the signal from species that are at low abundance. So while you get broad coverage because you retain everything, the fact that you retain everything in your sample is affecting the detection of those metabolites that are at very low concentration. So that's the reason why, depending on what is like very broad initial hypothesis of which sections of metabolism are interesting to your project, then that can help you tuning the initial sample prep as to minimize that in a way that you can favor the detection of that subsection of metabolism that is interesting to you. So it's really a trade-off. There is no one-size-fits-all approach. It's I would say this is a big difference of metabolomics across other omics sciences, across transcriptomics, across genomics, that have more unbiased coverage, or the discussion there is mostly in terms of depth of sequencing. Here is more about the chemical-physical nature of what we want to measure, and how the technique that we use for measuring that is biasing, is introducing a slight bias in what we can measure in the frame of that specific analysis. So this is an initial, so the sample prep already introduces, it's dictating what you're going to see later, right? But as in most of workflows here particularly, the next step is in the LCMS platform, so the chromatographic separation. The chromatographic separation can be performed by using different columns with different stationary phases. But why do we do chromatographic separations in the first place? So based on what I told you, anybody has an idea of why are we complicating our life to add an additional dimension of information? And you can, there is no scoring on this. So as we do have ion suppression, right? As more complex is the sample that we inject at the source of the mass spectrometer, by separating through liquid chromatography, we are able to simplify over time the composition of our sample. And that allows us to inject gradually different metabolites. So the mixture, even if it's not 1-to-1 at every time of the chromatography, but still it's gonna be way more simple as a composition than having a shotgun injection, right? Injecting everything at the same time. So that's one of the first advantages. One of the first advantages is that we reduce drastically suppression, ion suppression. However, there is also another aspect that is very important, and we use, and is informative in metabolite identification, And that is the fact that depending on the affinity for the stationary phase and mobile phase of our analytes in the chromatography, so while they run in the chromatographic run, this can be more or less retained. And that informs us on the nature of that metabolite, right? Because depending on their structure, they will either have more affinity for the stationary phase or the mobile phase. And that is an information that it translates, it's reflected in the retention time of that specific molecule. And ultimately, in the identification, but also in performing considerations pertaining what is the structure, what is the nature, which part of the metabolic map does this metabolite belong to, this is an information that is very useful in the interpretation and identification. Any question? Okay, so the next step is the mass spectrometer. In this case, this is global metabolomics. So we are performing detection in untargeted full scan, meaning we inject our sample after the chromatography. So at each time point, this injection is what we inject is a simpler, it's a simpler version of the overall sample that we are using. And what happens is that we need to detect it. We need to make it visible to them. In order to do so, okay.
Hello.
Yes. And Anadita, do you have a question?
Oh, sorry. No question. Sorry. No
Okay.
question.
Okay. So we need to make those molecules visible to the mass spec. So how many of you are familiar with mass spectrometry? Okay. What is the fundamental detective principle of mass spectrometric detection in ionization? Yes. Okay. So we need to ionize and separate and measure, right? And this is all done by the mass spec. So, but we need to prepare our molecules to be ready to undergo that. So the first step is ionization, and we do that in the source, which is usually an electrospray ionization source. Is a soft ionization technique, meaning that we aim and tend to obtain the molecular ion as opposed to breaking the molecules in bigger fragments. So the ionization can be performed in positive and negative mode, meaning depending on which type of potential we apply. So positive mode, the potential will add usually a hydrogen, so a charge of +1 on the molecular, on the molecule, on the molecular mass. The negative ionization will add an electron, right, or remove a hydrogen as from the perspective that you want to see that. But the final result is putting a charge on our analytes. And that is important because that is needed later on in the mass spec. In this case, this is a cutoff. To accelerate the molecules based on their charge and mass across two different potentials, and then measuring their time of flight. And that time of flight is proportional to their mass, but also to their charge. So depending on the mass, depending on the charge, or let's say if we put constant the charge, we will be able to separate these molecules based on mass. And that is informing us, that's the basic principle of how these molecules are measured. Eventually, we end up with a mass spectra. This is a total ion chromatogram in the top right corner, which is a sum of all the signal at every given time the detection is performed out of the LC. After this, there is data processing. meaning that we need to use algorithms to perform peak picking. So we need to have this data. Remember, we are in untargeted, so we do not know a priori what we want to measure, but we can use our expected knowledge on how the peaks of molecules would look like. So, and that can be predicted mathematically, and then it can be fitted through algorithms in our data as to extract the part of data that is consistent with what we expect for a molecule. So that is called peak picking. Retention time correction, right? So each one of these peaks that we will measure for a specific molecule, we have an associated retention time, which is the time at which the molecule elutes out of the LC. And that is informative on the molecule, on the identity of the molecule. Then we perform chromatogram correction and alignment, and that is because as you progress in a sequence, a measuring sequence that has n samples, particularly the chromatography, also the mass spectrometry, but way more so the chromatography, drift in their parameters. So that means that the signal has a systematic and consistent shift that can be corrected post-run. And that's what we do at that step. Putative annotation. So from these data sets, one that we made them, so we extract the data, we extract the peaks, we make them comparable across different samples by aligning and comparing retention times. That gives us a list of M over Z value, mass over charge values, a retention time for each one of those molecules, and abundances across the different samples. But at this point, we need to start understanding if we can annotate some of those molecules. And that is called putative annotation because it's not an identification, it's matching the measured M over Z value with databases of expected values for known molecules. And that provides, it's usually, depending on the accuracy of your mass spec, to some extent also the resolution helps, but it's mostly the accuracy of your mass spec. That matching of the measured versus the database value can give you from, I would say in a typical experiment, you can, and it depends a lot on the molecule too, but for the common ones from 5 to 20 to more than 20 annotations. So from that, you can get a sense, especially when you project that in specific pathways and you have changes that are very consistent across pathways of what the overall part of metabolism that is changing is. However, it's not an identification. So your confidence in your results is still preliminary and can just help you generating hypotheses. It needs validation later on in the pipeline. And then data pretreatment and normalization, right? So this is mostly what we do also in other omics approaches when we have a big data matrix. And then we use a univariate analysis, of course, what we use in every experiment. Pathway analysis, meaning taking this data and contextualizing them in the pathways. And as we will see later, we can do this in two different degrees of depth and using different levels of identification or annotation. And then also integrating this information in the broader context of the system level regulation, right? Proteomics, phosphoproteomics, transcriptional data, CRISPR-Cas9 screenings, and GWAS association studies. So really, there is a wealth of information that can be used to enrich the interpretation. So, okay, do we have any question? Okay, this slide is only to give you a visual of what happens in the chromatography, right? You have the sample, it's a complex mixture of analytes. Depending on the chromatography, the role of the chromatography is to separate this molecule. So you see that they separate in the chromatographic run and they will get to the detector, which in this case is the mass spectrometer, separated. So simpler to detect. And the foundation of this separation is based on the affinity of the molecules for the stationary phase, which usually looks as those spheres that you see in this column. and the mobile phase, which is the solvent, the mixture of solvents, and the gradient of how those always change while the chromatographic run advances. Right, so that's how it looks on, I would say, the millimeter scale on the left. On the right, that's how it looks operationally in the lab. You have probably seen these columns many, many times, and different types: reverse phase, C18, helic. So all these type of names refer to different types of mobile phases, and also the technology of the spheres. Some technologies are more advanced and better in resolution and separation, and resolving and separating the metabolites. In general, the one that is considered most advanced today for the lab routine is the ultra performance liquid chromatography, which has very small stationary phase particles. And but there is a lot of science there as well and advancement. So it's always good to keep an eye on how technology advances there. So the mass spectrometer, these are the main three elements of it, with an adjunct, more functional role of the vacuum pumps. So the ionization source, it can be, it depends. So the most common one, as I mentioned before, is the electrospray ionization for the source. And the reason for that is because it's very versatile and it's soft. So it's not causing the fragmentation of the compounds while you ionize them. Now, this has been demonstrated being too much of a big expectation, because when it was developed and in the first decades that it was used, we tend to think that that was the case. But over the last decade, we have clearly appreciated that even soft ionization approaches such as ESI cause in-source fragmentation, and that can complicate your interpretation of the spectra later. So that's something to keep in mind. Like, we would like to think that it's completely soft. It's definitely softer than chemical ionization or electron ionization, which are called hard ionization approaches, because their goal is to fragment the molecule really at step one, at the ionization before detection. So ESI is probably the ionization source that you will find the most in, and that probably you have in your lab as well. So then atmospheric pressure chemical ionization, this is a soft technique, it's less common. It works at atmospheric pressure, and it uses gases, chemical gases, to cause the ionization. SIMS is secondary ion mass spectrometry. It has a secondary stream of ions that causes the soft ionization. So these are all variations that has been developed over time. Plasma desorption ionization, right? Like, I'm very happy to talk more into this if you have this type of source in your lab and you are interested in understanding it better. For the sake of time, I think we can go ahead. So the analyzer is usually we find two types of analyzer: the time of flight analyzer and the Orbitrap analyzer. What is the difference here? So the time of flight is a linear geometry, is a linear structure. We do have, as you can see in the slide, see if this pointer works. Okay. So we do have the first part here. That is a quadrupole, and that quadrupole is there. You can imagine it as a filter. You can use it to let every ion pass, and that's usually what is happening in global metabolomics. But you can also set it to a specific range or a specific molecule that is also called precursor, in order to isolate that precursor and then send it later on to the detector, to the time of flight detector, right? So the time of flight detector is here. And that's really where you have this, in lab, you can see it's a long tube where your molecule enters. And based on the potential of different of potential, follows this trajectory as an arc. And depending on the mass, these molecules will separate in the run and hit the final detector at different times, right? So that's the whole principle of separation. Why do we have the quadruple present in this geometry? Like, why do you think in some instances, based off what I described to you, this quadruple might be needed, might be used? Like, why might we have a need at a point to isolate a precursor at that early stage before the detection? Yes. Yes, so in targeted approaches, for instance, yeah. But there is a need also in global metabolomics. Okay, so when you want to learn about the identity of the molecule, you need to fragment it, as we mentioned before. But how do you fragment it? You need to go back to the mass spec with your sample, and then you need to set the quadruple to be able to isolate that precursor. Because then, after that, you fragment it, there is a fragmentation chamber, and then you use the QTOF, the TOF part of the QTOF to detect the fragments, right? So that's why it's there. So there are some, not for QTOF, but for instance, you can have triple quads or even quadruples that were like the very old first version of mass specs. They will still work like independently from the QTOF, from the TOF. The problem is that resolution, accuracy, it's an older technology, but it still reveals useful to isolate. Or like, for instance, when you have a triple quad, which is 3 quadruples in sequence, even though that's not meant to be appropriate for untargeted, because the accuracy is not to the level of the TOF or the Orbitrap systems. When you are in targeted approaches, sometimes a triple quad can be better than a QTOF. And the reason is because it can get better sensitivity when you know what you're separating and what you want to measure. So it has more of that application. But it's a way older technology, and also it's the investment to acquire it is way more limited than a TOF, a QTOF, or an Orbitrap. Like with a QTOF, I think today we are talking probably in the order of $500,000 minimum. With an Orbitrap, which is a way more sophisticated mass spectrometer, it's usually at least in the order of $1 million or higher than that. So what is the difference with the Orbitrap? The way the ions are separated and detected is different in the Orbitrap. While here we have a linear geometry, the TOF, in the Orbitrap we have this fuse, right? And the difference of potential is applied between this part and the fuse inside the detection. And that what that causes is having the ions, the molecules, starting rotating around that fuse at different frequencies, depending on the mass and charge that they have. And that, provides very complex plots of frequencies of rotation that can be deconvoluted through what is called the Fourier transform. So it's still like if you think of it as a phenomenon that you observe, it looks very complicated, but because of the nature of the geometry of the fuse, it's actually very repetitive, and it has allowed to develop equations that are very accurate in the way they enable to dissect the frequency and the frequency of the oscillation back to the mass over charge ratio. And that's by far the most accurate instrument that you can have in the laboratory. So now, why is accuracy so important? Accuracy is important because while with a TOF, usually we can get in measuring mass over set values with an error of ±5 parts per million on the mass. With an Orbitrap, we can go lower than that, usually around 1. And when you use those masses in matching back to reference databases, that narrows down the number of annotations, meaning with a cutoff, you might have for that M-visit value a match with 20 molecules. If you measure the same with an Orbitrap, you will end up with 4 or 5 different options and work on those. So it's a big advantage. It's usually a better instrument for proteomics for many reasons. One being definitely the accuracy, but also the resolution that it provides in the peak, in the shape of the peaks in proteins and in peptides from proteins. And but like the peaks in proteomics are per se more complex and closer because of multi-charges. So that's a different discussion to some extent. But I would say Orbitrap, excellent instrument. If there is the funding to acquire it, that's perfect. But to a certain level for performing metabolomics, having a cutoff is usually very good and performs generally in terms of applications and your end result and goal. It's very similar to what you obtain with the Orbitrap. So this is a total ion chromatogram, right? Simply the retention time here, meaning this is every moment when that mix of
Amelia?
Yes.
Oh, I had a quick question about the slide before.
Yes, Sophia.
The slide before this one, just talking about the Orbitrap.
Yes.
Can you hear me?
Yes, this slide.
Okay, yeah, because on the Zoom, okay, it was slow on the Zoom. Could you explain again what R means exactly?
Yes.
And then again, kind of just what R means exactly in like the parts per million, because in our lab we use the Orbitrap and we can go very small for the parts per million, but what does that mean again, the ppm?
Yes.
Because I do it, but then I'm like, I don't really understand what that means when I'm selecting.
Yes, and we will have some, I believe we have some slide later that will allow us to look better into that with an example of with the actual peak of a molecule. Anticipating on that
Okay.
is resolution, right? Accuracy. Those are two different concepts. Accuracy is a measure of how close is your measurement to the actual value, and in this setting, how close is your M/Z, the measured M/Z to the actual M/Z of the molecule. And of course, the closer that is to the actual value, the closer you are and more specific you are in the annotation, right? The resolution is more a measure of, let's say, like how resolved are the mass spectrometric peaks that you measure. So if you have peaks, M/Z peaks that are very close in values, in the real values to each other, how good is your instrument in separating them? Or is it gonna give you like a combination or a partial combination of those peaks? And that is very important in proteomics, because in proteomics, you have multi-charged peptides to measure that make the peaks that you detect being apart from each other of fractions of 1 unit of mass. Like, for instance, you can have 0.5 distance, 0.25, right? Like fractions of that, depending on the charges. And in that case, you really need resolution to resolve those clusters of peaks. In metabolomics, that is important to a certain extent, because you usually have peaks in that describe a molecule, a metabolite, with a 1 unit of difference, right? You will have the molecular +1, the molecular +2, the molecular +3. And for that, you need some resolution, but I would say the resolution around 10, 20,000 is already good for that. Does it answer your question?
Yes, it does. Thank you. Yeah, because a lot of times the difference between the molecules is so small that it has to be able to scan for that, especially with amino acids.
Yes, yeah. So that when you start stepping towards protein peptides, and so that's where you start having an advantage of using Orbitraps. For some lipids as well, it's an advantage too. Of course, it's a top-notch instrument. So if you can have that, and that it usually performs very well for metabolomics as well. Okay, any other question? Yes.
So I've used with a core multi-top before, and I was wondering, because I don't think, because that's like a fixed slice of tissue, so I don't think it has the chromatography piece.
Mhm.
But I know you can like filter to look at like proteins or look at lipids, like look at specific classes of molecules. Like, would that be where the coil comes in to do that, or?
Well, we would have to look into your specific setup a bit better. From how you described it, my sense it can either be at the level of selecting the mass range that you focus on, because lipids are more higher mass range. Proteins are, depending on how many charges they acquire, they can fall lower than that. But small molecules, they are usually from 80, 100 to 1000 or 700 even. So 700, more than that, is usually more lipids. So it really depends on the setting on your mass spec. Yeah. But it can be that, it can be what type of you don't have chromatography, you said.
I don't think so. Yeah, because it's just it's a fixed slice of tissue, and then I don't know exactly what it does, but it's like the laser capture.
Okay, that's called you use a laser for ionization. That's called multi-ionization. Yeah, which is different than the ESI. Yeah, so it's a different source. It's a more imaging. I can add more examples and information in the coming days about that. Yeah, because I have some more that we can expand in the next days. But it's more niche application, and it comes with its advantages and caveat when it comes to identification, because you don't have a quadruple that you can use because of the setup, right? You cannot use a chromatography in between because it's direct from desorption from the sample. So yeah, we can talk about it in the coming days. Okay. So this is the total ion chromatogram. For each time point of the chromatography, it shows the total current that is generated by the detection of the molecules, the ions that hit the detector at that time, right? So now this retention time informs us on the affinity of those molecules, meaning that the ones that we find at the beginning, also this part is also known the void volume, right? Which it's basically the part of your sample that is not retained and goes out of the chromatography immediately. This is low affinity to stationary phase. It's either not retained, very low affinity. The more you move towards this direction, the higher the affinity to the stationary phase, right? So here you find things that are very well retained. Sometimes you see here, these are likely things, for instance, lipids that attach to the column very tightly, and they might even not be completely flushed out. They might be accumulating in your column. And that's, in the quality controls across your columns, that's one of the aspects that is really important to look into. If your gradient, your setup is not optimized for the applications, the length of the chromatography, so a lot of parameters around that that allow you to make sure that your sample was in here and was out by this time, and you don't have further retention that is gonna be built up in the next round, right? And this is a typical one from untargeted analysis. So now let's look at this in 3 dimensions. So the retention time is here, the x-axis that we had before. The y-axis, the y-axis, the intensity is here. We had it before. So before we were looking at it from this angle. Now we expand it in this other dimension, that is the m/6 value. And then if you slice it, right, by each retention, specific retention time, then you can see which molecules are measured at that specific moment in the mass spec with the peaks and so on. So this is just for you to orient yourself into how is the actual structure of this raw data. So, and this allows us to, we say, through the convolution of the signal, finding metabolic features that are characterized by, we say, M/Z value. We usually see them in ESI as molecular in positive molecular plus H, right? Or in negative molecular minus H. Then we have the retention time, and then we also have something which is called the adducts, just to complicate everything. So the adducts, what are the adducts? Anybody knows what the adducts are? Okay, so our molecules are in a sample, and this sample is injected in a mobile phase through the chromatography, right? So both the sample and the mobile phase contain sodium, potassium, sometimes they contain ammonium, sometimes they contain other, like formic acid, other components. These are atoms or components that can easily complex our analytes. And some analytes are more keen to form these adducts, this complex, with elements from the mobile phase or the sample, as opposed to others. And that is important because some molecules are more stable in their adduct form when they are ionized, as opposed to their molecular form. So you will measure them, you will see them shifted of a mass that is, for instance, in the sodium case, the weight of sodium. Or like if it would be formate, it would be the weight of formate, and so on. So we do have, as we said, different chromatographies. We have an example here. This is the sphere in the chromatography that we saw before. In a C18 column, we have a hydrocarbon chain of 18 carbon atoms. It's meant to be a hydrophobic surface, and therefore it interacts better with things that are alike, right? More hydrophobic, hydrophilic. Usually this is called the helic. That's the most used one. In positive, negative ionization mode. The aminopropyl one is very common. It has an aminopropyl group as functionalization. And the difference is in that that's more polar. Therefore, it's more fit for measuring and retaining, for instance, central carbon metabolites, right? Like this one is has broader coverage, but the coverage is more biased towards things that are more similar to its hydrophobic nature. And there are many publications that give you a more that can chart that coverage, and based on what you want to measure, you can refer to that and start having an initial idea of where to start from, or at least before you buy the column for that and investing usually $1,000 on a column or 800. That's the price range. You can use that information to operate a decision that allows you to separate your components. So on top of this, the analytical toolbox, there are three main pillars that really enable global metabolomics. One is analytical chemistry. We went through that. Then we have spectral databases such as the METLIN, but also we will see later there are many others that contain this information about spectra and information on known molecules for a very, very wide range of metabolites. And also the bioinformatics around it, right? That allows to combine this information and finally extract data. So molecular databases, the biggest one is METLIN, but there are others that are available. The NIST databases, the MONA, the MZ Cloud, GNPS. GNPS, I believe today, this is 2020, so today it has been updated significantly. What they contain: the putative M/Z value, inserts fragmentation information. Fragmentation meaning the MS2 spectra that you can use to identify your molecule. You can compare that from the database with your molecule. MS1, MS2, and retention time comparison with pure standard. So you can find that too. And this information is reported gradually as reflect different levels of known metabolite identification, right? If you can have a very putative level 1, then if you add in source fragmentation, you can increase confidence, but still it's not identification. MS2, by comparing your MS2 from your molecule to fragmentation patterns in databases, you are very close to identification, but you are not 100% identifying it. When you have all the information plus comparison with a pure standard that you measured on your platform, then that is considered full identification. But there are also unknown metabolites, right? For these ones, NMR also provides additional complementary information. Cryo-EM is also has been used in a very impressive way to visualize, like to characterize the structure of small molecules as well. So of course, it's very expensive technique so far, but very, very powerful for looking into this kind of structures. Synthesis and analysis of pure compounds, right? So, for instance, for lipids, I've seen like over my projects and work, sometimes there is a lipid that is inferred in terms of structure by the MS2, but there is no standard available commercially. So what do we do in that case? We either switch to another approach that is not mass spec-based, or we try to synthesize our molecule, the one that we infer is the identity, and then we measure that in our mass spec. But in that case, still we will need also for our synthesized compound an independent method that confirms that identity. So I would say this part is very it's not time consuming. In source fragmentation, there are tools that allow to integrate that too in this first step, so not time consuming. This one, there are several options, is slightly more time consuming, but still accessible. When it comes to this and this part, it can be from months to years, depending on the molecule. And some molecules have been, like sometimes it's just so difficult that one prefers, to invest time on different type of questions, or sometimes it's not relevant. It's more important to characterize the pathway and then seeing whether that molecule changes as part of that pathway. So it's really here the consideration of how much of my time am I going to invest in this. It's really important how many resources I have to really go through all of this, because that's resource consuming. And for lipids, there is LIPID MAPS as a reference database that is focused on them, while the ones in that chart are mostly focused on the small molecule fraction. They have some lipids, but the gold standard for lipids is LIPID MAPS. Here you find the links to them if you're interested in looking what type of information you can find there. And this is my last slide before the break. So we are running a bit, so let's go briefly through this slide, and then I think we will take 10 minutes instead of 15 for a break. So these are metabolomics data repositories. Now, metabolomics is one of the omics sciences, right? It's the youngest one. So genomics was probably the earliest one, transcriptomics as well. We do have gold standard repositories for that. We are all very familiar with them. Metabolomics has repositories as well. They are open source. There is data. We will use this data in the second part of this workshop today. We can reanalyze them. We can do meta-analysis on those platforms, but they are usually less known. But they are expanded. So the one that contains more accessible information and is based in the US, in San Diego, is the Metabolomics Workbench. We will navigate it later as part of the hands-on training, but there is also Metabolites and Metabolomics Exchange. Metabolites is more a European effort, and Metabo Exchange is more a data aggregation website that allows you to search into different repositories, particularly for those repositories that have raw data on plants. I believe the GNPS now also includes some of the raw data, so it's always an evolving scenario, but these are the ones that I tend to consider first when I look for data. Another source of raw data from other studies, of course, as in transcriptional studies as well, is literature, and just contacting labs and seeing whether they want to share. We should consider that untargeted metabolomics data are really information rich. And generally, they are analyzed to answer a specific question in a specific comparison. However, that information that they contain is not completely distilled in the frame of that comparison, and it's useful to many other meta-analyses. So that's similarly to transcriptomics and other omics sciences, right? So it's nothing different here. Okay, so if we don't have any question, I would take a 10 minutes break and we can resume in so now it's 10:28, we can restart at 10:40.